Welcome back to the lecture series for class 12th. In third and final part of human reproduction, we'll learn about fertilization and implantation. Let us look into what are the steps involved in this process. It begins with insemination, where the sperms are transferred from male to female parent. Sperms being motile, they swim rapidly towards uterus, ultimately to reach fallopian tube. By the time sperm reaches ampulla of fallopian tube, if the process is synchronized, ovum is also released at the same time and is picked by the fimbria of fallopian tube. Fertilization, that is syngamy or fusion of male and female gametes, take place in the fallopian tube. This image represents the path of both male and female gametes into the female reproductive tract. This blue arrow represents the path of sperm and the purple arrow here represents path of egg. Sperms, after entering into female reproductive tract, it reaches uterus and eventually reaches the fallopian tube. At the same time, if process is synchronized, ovum is released by ovary, is picked by fimbri and reaches to the ampullary isthmus junction where the fertilization takes place. Fusion or syngamy of male and female gametes takes place. Here you can see that all the stages of fertilization and implantation of blastocyst is shown in a single image. It begins with the processes taking place inside ovary and how fertilization takes place, what are the post-fertilization developments and changes, and how it results into implantation of the embryo. So let us have a closer look. Inside the ovary, primary follicle matures into secondary follicle and then into Graafian follicle, which ruptures and secondary oocyte surrounded by granulosa cells is released outside. Rest of the granulosa cells matures into corpus luteum, which secretes progesterone and is required to maintain pregnancy and to facilitate implantation. Secondary oocyte, after being released from ovary, is picked up by the finger-like projections known as fimbri of the infundibulum part of oviduct. It reaches to the ampullary isthmus junction, and at the same time sperms also reach to the same junction, and in the due process of their moving towards the oviduct, they get activated through the female genital secretions, and this process is known as capacitation. Now, at the ampullary isthmus junction, when syngamy has to take place, a sperm will contact zona pellucida of the secondary oocyte and will induce changes in the membrane so that entry of other sperms are blocked, and it is ensured that only one sperm can fuse with the oocyte. This is facilitated with the help of some of the acrosomal secretions which help in penetration through the zona pellucida layer and the plasma membrane. Some of these secretions are hyaluronidase, corona penetrating enzymes, and acrosin. After penetration of a sperm through the layer of zona pellucida and plasma membrane, now the meiosis II is completed in secondary oocyte, which is an unequal division and results into a large single ovum or otid and a small tiny second polar body. This ovum is now available to fuse with the male gamete or the sperm, and fusion results into formation of zygote. Zygote undergoes successive cell division known as cleavage. Cleavage is holoblastic in humans. Cleavage results into formation of 2 cells, then 4 cells, and then 8 cells, and so on. These cells are known as blastomeres. Ultimately, the cells divide and reach 8 to 16 cell stage, which is known as morula. Following the formation of morula, it gets developed into blastocyst. The blastocyst moves further in uterus. At the late blastocyst stage, now zona hatching takes place. This is a process where zona pellucida layers are removed. Actually, zona pellucida layer prevents implantation of blastocyst at any abnormal site. You might have heard about ectopic pregnancies where implantation takes place anywhere outside the uterus, mostly in the fallopian tube. This is because the trophoblast layer is sticky and is exposed to the site, to the abnormal site. So zona hatching may have taken place earlier in those abnormal cases. Normally, the zona hatching takes place during the 6th or 7th day when zona pellucida layers are removed and the sticky trophoblast cells are exposed so that they can stick and get embedded into the uterus. This results into implantation of the embryo. A small revision of what we have seen earlier. It begins with a sperm contacting with the zona pellucida layer, a single sperm, so that it can block the entry of any other sperm. So this is to ensure that the process of fertilization results into a single offspring, a single fetus. Now, for this penetration, we need acrosome secretions that I told you. There are three major secretions: hyaluronidase, corona penetrating enzyme, and acrosin. They penetrate through the plasma membrane and zona pellucida layer and ends up into meiosis II of the secondary oocyte, so that second polar body comes out and a single large ootid is formed. Now, fusion between sperm and ovum takes place, which results into zygote formation. Zygote undergoes cleavage, holoblastic cleavage, and results into 2, 4, 8, 16 celled blastomeres. Embryo with 8 to 16 cells is known as morula. It undergoes mitosis and forms blastocyst. The blastocyst, it has got two layers: the outer trophoblast layer, that is sticky, and inner cell mass. Trophoblast is the outer layer, it's sticky, and it is supposed to get attached to the endometrium so that implantation can take place. Uterine cells divide and cover the entire blastocyst. The blastocyst actually has to embed inside the uterine wall. So this is known as implantation. Inner cell mass differentiates into embryo. Next is pregnancy and embryonic development. After implantation, that is 7th day after the fusion of male and female gamete, there is development of chorionic villi. These are finger-like projections on trophoblast that is surrounded by uterine tissue and the maternal blood. Placenta, you might have heard about placenta. These are very important structure for the developing fetus. It is actually the interdigitation of chorionic villi and uterine tissue. So placenta also gets developed after implantation, and it is very important structure, very helpful for the developing embryo because they help in gaseous exchange. So these are responsible for the supply of oxygen to the embryo. These are also involved in removal of carbon dioxide and other waste material from the embryo, also involved in transportation of all other nutrients that are required by the developing embryo. Not only this, it also secretes hormones like human chorionic gonadotropin HCG, that this hormone you might have heard about. The pregnancy kits that also uses this particular hormone as a detection test of pregnancy. So placenta is also involved in secretion of HPL, that is human placental lactogen, and relaxin, which is secreted. These three hormones are secreted only in pregnancy, and relaxin among them is released only in the later stage of pregnancy. All other hormones like estrogen, progesterone, cortisol, prolactin, thyroxine, they are all increased during pregnancy. After implantation, the blastocyst contains two layers of cells: the outer trophoblast that is required for implantation to take place. It gets embedded inside the uterine wall, and the inner cell mass that further differentiates to form the entire embryo. Now, In this image, you can see that the maternal blood pool is seen, the endometrium is seen into which the embryo has been embedded. Uh, this embryo is covered with four membranes. The outer one here is chorion, that is derived from blastocyst cavity membrane. We have allantois, we have amniotic cavity containing amniotic fluid. The structure is known as amnion, and then we have the innermost yolk sac which is lined with endoderm. So inner cell mass is required to get differentiated into the different types of cells, and overall it forms the organ. Inner cell mass gets differentiated into the outermost layer ectoderm, middle layer mesoderm, and the innermost layer endoderm. Endoderm further gets differentiated into the cells of internal organs such as cells of lungs, the alveolar cells, or the digestive tract or the liver, etc. Mesoderm, middle layer, it becomes the cells of connective tissues or the muscular tissues. All sort of muscles like cardiac muscles or skeletal muscles or the smooth muscles, they get differentiated from the mesodermal layer. Similarly, all the cells of connective tissue, they also get differentiated from middle layer. Ectoderm becomes the outermost part of the organism, like it gets differentiated into skin, the nervous system, nail, hair, pigment cells, etc. Next is parturition and lactation. After the 9 months of gestation is over in humans, it is now the time to end pregnancy and expel the fetus out. So there are three stages. It is end of pregnancy, that is for which the signals are originated from fetus and placenta, and it results into induction of uterine contraction. Second stage is the contractions to be growing stronger and stronger. So following the signals from fetus and placenta, the neuroendocrine mechanism of signal transduction, it induces only mild contraction of uterus known as fetal ejection reflex. But oxytocin also gets released from maternal pituitary gland, which induces stronger contraction. And there is a feedback mechanism. It acts in loop. So oxytocin inducing stronger contraction again stimulating pituitary to release more oxytocin, oxytocin further stimulating, further inducing stronger contraction. So eventually contractions grow stronger and stronger and ends into expulsion, that is parturition. The baby is delivered through the birth canal and placenta is also expelled out. That's all in this video. Next chapter is reproductive health. Stay tuned and please subscribe our channel. Thank you.